are live on my Facebook. Pat, it is a pleasure to have you. We'll go through a couple of introductions. I've labeled this podcast, actually, it's episode 14. If you can believe it, I started these during the quarantine, and I've had a couple a week and then sometimes just one a week. And I've labeled this one From Scientist to Entrepreneur to Executive, the story of the never be outworked mindset, which is the theme of this podcast. And you just exude that example. So we'll talk about more about that later. So you're listening to Secrets of Success podcast with Dr. Shane Needham, where the theme is never be outworked. You can follow me on Facebook. You can follow me on Instagram. You can follow me on LinkedIn. Visit my website at drsneedham.com. All these are recorded live on my Facebook, streamed, and also they're recorded, and then they are available about a week later on YouTube, on my YouTube channel. You can find that just via Google search, on Spotify, on iTunes, or on SoundCloud. And I am honored and very content and happy to introduce Pat Bennett. He is the VP at PPD, and we'll talk about that in a minute of what that is, but he's in my industry. So I own a pharmaceutical testing CRO, and he is part of an executive in a pharmaceutical testing CRO. So we call ourselves competitors, I suppose, but I look at it as
really.
I look at it as collaborators. So it's really, really good. I've known Pat for many, many years. I knew Pat before I started Alturas Analytics. And so we are taking callers if anybody wants to call in. And we are taking callers at 877-995-2111. I'll give that number out. Again, I might even put it on the actual stream if I can do that quickly without being too much of a distraction. What I'm excited about is I've already talked about this, but why did I start a podcast? And so I've been involved in science for over 25 years now. I've been wanting to be a scientist since the age of like 12. So I'm living the dream. It's really, really cool. And I know Pat feels the same way. He's got such a positive mindset. And so over the years, how we've disseminated information as scientists is we actually go to conferences, present a poster, present an oral presentation, which is very crucial and important. And I love doing that stuff. And or we write what we call a white paper for our industry, where we do some research and have a hypothesis, use a scientific method, write it up. It gets reviewed by peers in our industry, and then we submit it again and resubmit it and revise it. Sometimes that process can take months or years. And the reason I started a podcast, I think it's great timing during COVID-19 and quarantine. It just kind of happened to be that way that I started it then, because I've been thinking about it for a year or so. Is that we need a better way as scientists to get our voices of science and what's happening in the industry out to the global community of non-scientists as quickly as possible. And frankly, I don't think in the scientific world we do a very good job for many reasons. There's lots of intellectual property reasons, confidentiality reasons that we don't do a good job in utilizing social media. And here's what I know. If you are probably under 30, and Pat has children and grandchildren, you're not reading white papers very often. You're looking at social media, 1 minute clips, 2 minute clips. And that's how what part of this podcast is. I want to start disseminating that information in that fashion. So not white papers, but immediate disseminated information, live too, where you can talk to a scientist. And another thing I wanted, and Pat and I talked about this offline, is that scientists are normal people. We might be strange. We are probably, but we're not untouchable. And so we can answer anybody's questions from the non-scientific community. So without further ado, I really want to introduce Pat Bennett. He's a vice president of PPD. He'll talk a little bit about that. He's been in the bioanalysis world, the scientific world for a long, long time. And so Pat, I just want you to introduce yourself and, tell us about yourself. Go back as far as you want, be as personal as you want. And we really want to hear your story. And Pat doesn't have a typical story necessarily. It's definitely a never be outworked mindset. So let's talk about that, Pat.
Yeah. I think I've always been that way.
You have.
Don't ever remember not being that way. Maybe it's because I was the second child and needed to keep up with my older brother.
That might be it. Yes.
And I was tiny, man. So yeah, I always wanted to play with the big boys, even though I was small. Yeah, well, the not typical is absolutely correct. Sometimes I used to tell people if they're the right age, they remember Green Acres, right? The TV show, but most people won't remember that. That's how I grew up. I spent a lot of time on my uncle's farm. I was born in the country up northern Pennsylvania in Bradford County, PA. Then moved down into Long Island, right. I could climb the trees and see them building the trade towers from outside my house. That's how close I was to New York City, right? So summers and winters in the up on the farm, hanging out with my cousins, just loved that. And the rest of the time I was down in the city, right? So I had both. I understood country and I understood
Wow.
city.
Wow.
So that was pretty interesting. And then growing up, I didn't expect to even go to college. Just that wasn't really the way that we did things. We were general contractors. We built houses and things like that. I wanted to do custom auto. I love
Wow.
cars, loved custom
Cool.
auto. Right? Yeah, that's what I wanted to do. But timing just happened to work out that, and wrestling. We'll talk about wrestling later.
Oh, I can't wait.
But I wanted to keep wrestling. And so I figured, all right, let me go to a good school if I'm going to go to school, let me go to a good school where I can wrestle too. So I just went to the local community college. And kind of what drove me towards what I did was wrestling. I was hungry. I mean, not just mentally, I mean, physically, I was hungry.
Absolutely.
you know
Yeah.
and in order to avoid food, I'd go to the library, and this is how I started thinking about science. I'd go to the library, and read about nutrition, right? How can I learn about what little food I'm going to eat? I need to make every ounce of it, right? And
Absolutely.
so I'd
Yeah.
go to the library and read about nutrition, and this long-haired kid would walk in the library asking for books on nutrition. The librarians would just like, you know, freak out, like, what's up with this kid, And I'd be in there all day long, hours, just to avoid eating and learning. So that's what got me going. And I thought, okay, I want to do something with food. Looked
Yeah.
at nutrition and said, nah, that's not a good career move. I can't see how that works. Thought about being a chef, and I really wanted to be a chef. But the school that I picked out, Johnson and Wales, wasn't matriculated at the time. Now it's a university, but it wasn't matriculated,
Interesting. I
which
didn't know
meant
that.
that I got a BS, but I didn't have a BS. I could not go to anywhere else for any other degrees. I'd have
Interesting.
to start all over.
Because
So
that's a really good school
yeah,
too.
yeah, it would have been great. I think I would have been awesome. Yeah, would have had a
Interesting.
lot more tattoos if I went in as a chef.
Yeah.
But
That's
so
so.
anyway, go ahead.
So what weight class did you wrestle at in community college?
118.
Okay,
Yeah.
of
But
course.
I'd
Yeah,
go in before practice. I'd go in weighing like 130 and come out weighing 118.
I know. Only, and I find that it was when I first met Pat. We'll get into that in a while. And there's several people in our industry. I won't name them just
Yeah.
but you see them at a conference and they're scientists, and you look at them, it's like, he's a wrestler. And sure enough, you're like, did you ever wrestle? Yeah, I did. How do you know? Anyways, and
The way we hold ourselves.
yeah,
Yeah.
and it's a different mindset, and I think it's great
Yeah.
to be surrounded by people like that. What a great story. So
Yeah.
you chose your local community college. Now, what was that? What was that college called?
Nassau Community College
Okay, and so that
on
was
Long
on
Island.
Long Island,
On
right?
Long Island. Yep.
You
Yep.
got it.
Yep.
And they did. So they had a wrestling program.
Yep. Yep.
Were you scholarship there then?
No, no, no. They didn't have scholarships. It was the 1970s. I was below poverty level, so I didn't pay to go to school anyway. Not
Yeah,
not in the beginning. Not in the beginning, right? We'll get to that part later.
yeah, because you're not poverty level anymore. You, by the way, I am. Yeah, it's just so cool to see people that have lived the American dream, and you've truly done that. And
Yeah.
I think we still have people today that say that that's not possible. And I'm not going to take away from your story, Pat, but it sounds so familiar to mine. Working on
Mhm.
farms, first generation educated college, and life is what you put into it. You
Yep.
get what you earn. There's no entitlement. There's no deserving. It's what you earn. And it's this guy. I mean, Pat is a grandfather, and he outworks everybody that I know. And we'll probably get into that as we go. But anyway, so continue. You go to
Yeah.
community college. You
Yeah.
rest.
So, do you know what my first degree was in? Hotel restaurant management.
Is that right?
Yeah. So
Now, was that
I
like an associate's degree then, or
uh-huh.
something?
Yeah,
Okay,
yeah, yeah,
that's
yeah.
awesome.
So I worked in kitchens. I was a cook
Wow.
while I went to school. Yeah, so all through my bachelor's. So I did eventually go in after I finished that degree. I realized I just wanted to be something different than in the restaurant. So
You know, the good part about that is you could pay for school while you're cooking and so on and
oh, it
so
was
forth.
great. It was great. I really had a blast doing it. It was great. And so then I went to St. John's University College of Pharmacy and into the toxicology program. So I got my bachelor's there and continued working in the restaurants while I did that. Then it was a recession, so I said okay, and I wanted a PhD, so I said, all right, let me just keep going. So I went into the master's program in pharmacology, and it was a great program. I met some great professors, great people. The laws changed though, and all of a sudden I got hit with a $30,000 tuition bill out of the blue. So I had to change everything that I was doing. Yeah, so I had to quit the restaurant and go get some other. I had to become an assistantship in the university, so I get the yeah. So yeah, it became pretty tough, but then I started working in. I realized I need a real career moving job, right? This restaurant, this assistantship. So I managed to. It's one of the things. Also, you talk about hard work. The other thing is luck. You have to create your own luck. It's
Totally.
not just hard work. There's a mixture of luck, but you have to create the luck. Recognize when it's there.
Well,
And
you
I
know,
went.
I'm not surprised that you create your own luck. And I want to be clear to people who are listening here and who watch this is that it's luck, but it's not. And
Mhm.
what luck is it's when preparation and opportunity meet each other.
Yeah, yeah,
So
exactly.
there's some people that are not afraid to take an opportunity, but
Mhm.
they're not prepared. They
Yeah,
don't have the education, the background, the experience, or whatever. Then there's a lot of people that have all the background, all the experience, all the education, but they're afraid to take risk, and
yeah,
they don't want to take an opportunity. It's
yeah,
like stealing second base. That's an opportunity, and you have to prepare for it. And so anyway, so I just I love that you're talking about you create your own luck,
yeah,
but I want to make sure we talk about what that is because
yeah, yeah,
you're a lucky guy because you're a thankful guy. I get that, Pat, but you created it yourself
yeah,
because of
yeah,
preparation and opportunity.
yeah,
Okay,
yeah.
so
You do.
so
So
go.
so what happened was I started working at Smith Kline, a toxicology lab. So I worked full time, and then I went to college for my master's full time. And that was, it was great that the lab let me do that. My operations were I worked 4 to midnight, and I'd work the weekends, and yeah, it was really good. Pretty fortunate. But then my we got Forrest came along, my son Forrest, and I said, okay, I don't want to live in New York City with and raise a family here. So I took my master's and went up and got a job at Bristol Myers. It wasn't Squibb at the time, Bristol Myers up in Syracuse, but still knew I needed to do something more. So after I was there for about a year and got my feet underneath me, I applied for an MBA program at Syracuse University and then went there.
That's
So
awesome. So
yeah.
why'd you decide? I have so many questions for you. We'll just let it flow. It's awesome. I love this story. I just love it. So why'd you decide to not pursue the PhD at that point and with
Yeah, you
the
know,
MBA approach?
so it's a combination of things, and I don't know because I can never be honest with myself about it, right?
Yeah.
So
Yeah.
some of it was, am I going to be the best PhD out there? And I'm like, I'm not going to be, right? So I'm like, well, okay. So if I'm not going to be the best PhD, how do I do this? And when you're going through those programs, it's really academic, and that didn't interest me. So how do you make that jump from academic to I'm doing something, right?
I,
So
I totally get it.
yeah.
That's why me with a PhD is like a fish out of water.
Yeah. So I decided no, I'm not going to do that. And I said, but the thing that I know I can do is I can marry that the science. I can marry that with the business, and the business was inborn in me. My grandfathers, my father. I mean, my brothers. Everybody understands business, so that was inside of me. The marketing. We used to talk marketing and through commercials, we'd answer somebody and we just do it with a commercial, so just,
I love it. I love it.
So
Yes.
I figured that would work, right? That
Yeah.
marriage, right?
Yeah.
It would really work, and it has. It absolutely has, at least
Well,
for
you've
me.
done a great job with it, and it is it's a skill that I think we're learning that it's more and more important.
Hmm.
Is because a lot of times, you know, you would come out of graduate school with a master's degree or a lot of times a PhD, and you knew nothing about management, you knew nothing about finances, you knew nothing about balance sheets, forecasting, any of the productivity, efficiency, none of that. Now I
Hmm.
think we're finding that that is just as important or more important in the world that we live in, because
Yeah.
you know the days of you have as much research budget as is necessary, those days are gone,
Oh yeah,
and
yeah.
they're not
It's
coming back.
brutal. Yeah,
They're
it's
not.
brutal.
I mean, we lived in the pharmaceutical industry during those times in the 90s. It was awesome. You could order anything you wanted, anytime you wanted.
Yeah.
That's not coming back. What are your thoughts on that?
Yeah, no, absolutely. You're 100% right. And that's actually, so we come back to creating those opportunities and that luck, you know. I, while I was working on this, I also had the opportunity to buy the second. Am I allowed to say brands?
Yes, yes.
Okay.
I talked about brands last week, so yeah,
All right. So
absolutely.
I
We'll get
bought
we'll get a shameless plug in there, Pat.
All right, so I bought the second SciEx mass spectrometer LCMS system in New York State. Jack Kenyon had the first, I was the second, right?
That's awesome.
And he happened to be coming up for a consulting uh, at Bristol Myers while I was there, and he heard about this other guy having the second SciEx. So he spent a little bit of extra time with me, talking to me, and
Wow.
2 months later, just by chance, I kept looking in Sunday papers to see what's the next trend. Where am I going to go for a position? I wasn't looking to leave Bristol Myers, but I was like, well, I just need to keep up on the market space. And flashing through the paper, all of a sudden the letters LCMS came up as I'm flashing through API LCMS. I'm like, what could, now come on. And so I take a look. It's in with assistant, admin assistant, and all of that in the A's, and it's about the size of my finger. That's the ad, 3 lines. And I look at it, I'm like, this is written for me. And I show it to my wife. She goes, that's written for you. I show it to my boss, and he goes, oh no. It was written by Jack Kenyon for me. And it's in their archive. It's in the Adbeyond scrapbook now, that ad.
Is that right?
Yep, yep. So
Wow.
that's how I left Bristol Myers, was that little tiny ad, just by chance, and
And so
me
then you went
getting
and worked
invited.
for Jack's Lab for a
Mhm.
while.
Yep.
And how many years? Because then you started your own thing as well. So
Yeah, so when
yeah,
we went there, it was just a mass spec and some pipettes. There was there was
I know
nothing
that
else there.
feeling. Yeah.
Yeah, yeah. So I was there, let's say May 93 until May 97.
Okay.
And then I had to do, you know, you get emotionally tied to these companies, especially when, you know, it's your blood, your sweat. I mean, literally, my kids would sleep on the floor and by the mass spectrometer on the
Wow.
weekends. We'd all go down because I lived in Syracuse, so it was a 60-mile each way trip. And
Wow.
so we'd work on the weekends, as you did too when you first started.
Absolutely.
And so
Absolutely.
they'd come down and they'd be running out with lab coats and gloves and stuff like that, you know, and they would literally sleep on the floor next to an API 3, you know.
Wow.
And
Wow.
so yeah.
Wow.
So but I let, you know, so you get tied to that, and so I wanted to respect the non-compete, so I joined two other people that had left Roche, and we started another company, and did you know, consulting for a year. That was awesome, but I couldn't say no to anything. I'm working my butt off, just, you know, okay, I'll do that, yeah, I can do that, I can do that. But I learned a lot from that too. I learned a ton about myself, about the industry, and about business from that consulting, you know.
Yeah.
So that
Yeah.
was good.
Because I think that, I mean, might as well get into that. Is I think that's when we first met, is you
It might have been with
it,
John Januszewski.
yeah.
Yeah,
Yeah.
yeah.
You guys came to Pfizer. I was there
Mhm.
probably with 98,
Yeah,
97, 98.
it might have been then. Yeah, that's right. That's right.
Yeah.
Yeah,
Yeah,
yeah.
and because he had worked with you at Bristol, and
Hmm.
then you went to Pfizer, and he told me a little bit about your story. And I was like, wow, I want to meet that guy. And so
Yeah.
I saw you run around in there, and so anyway, but yeah, just a small world. But so then you started the consulting thing, but it kind of morphed into something else.
Yeah, we knew that would happen. Once the year was up, that was it. So the choice was to continue to build a lab on our own or to do it with someone else. And my partners didn't have kids. I had two kids, and they were about 8 years old, and I had already moved them twice. From Syracuse down to Landenberg, Pennsylvania, and then over to Bucks County, PA. So they were in their third school in 2 years. So I said, yeah, I don't think I want to do this alone. So we basically sold that company to what became Tandem Labs out in Salt Lake City.
Got it. So your consulting morphed into also, but you had some mass spectrometers and everything.
Yeah, we built a lab in New Jersey that became Tandem Labs New Jersey.
Yeah,
in West Trenton, New Jersey. Yeah.
So what? Now you decided to sell because you didn't want to move your kids again. That was kind of the thing.
It was the risk, right? The question came to us from the CEO. Said, all right, do you want to be a big fish in a little pond or little fish in a big pond? And the response was big fish in a big pond. But I'm okay starting small. So that's what we did, right? That way it was broader. It wasn't just, the three of us here in this little laboratory and, two mass specs. It was, a lot more behind it, a lot more vision, right? It allowed us to do a lot more quicker. Yeah.
Yeah. When you merged with, what were they called back then? It's escaping me.
So it was NWT, which originally was Northwest Toxicology. Then they started NWB, Northwest Bioanalytical. And they also had a drug testing, NDT, Northwest Drug Testing.
Wow, I didn't know that.
Yeah.
So you merged your New Jersey lab basically with the Salt Lake. I'm trying to kind of explain for people who don't know, but your New Jersey lab with the Salt Lake City lab, and you guys became Tandem Labs, and the rest is history. And now they've been sold as well, and that's just what happens in our industry. And what a great story, Pat. It really is. I mean, and so. I want to ask this question because I get this a lot. I do have my own children, actually college age right now, and you hear you can't work at the same time and go to college. Do you ever hear that, Pat? Nowadays, I'm asking, do you hear?
Come on, man. Yeah.
Come on. So you're telling me you worked full time and went to college, and then you in graduate school you worked full time and went to graduate school.
Yeah.
Well, it sounds so my story
and started a business and started at, Advion. So 60 miles from my house.
Yes. # no excuses. That's what I have to say. I mean, what a great story. And I mean, mine was the same. I paid for my own college by working. I mean, I was a janitor. I worked in restaurants. I did anything, worked on farms, anything to make money.
I cleaned offices in high school. After wrestling practice, I'd go on over and clean a big insurance company or a bank. Yeah.
It's awesome. And it teaches you that mindset, even when you get educated, that it's not about your education. It's not about your experience. It's not about your intelligence.
Yeah.
It's about how hard you work. And that's one reason I took my first job at Pfizer is I knew that they had tuition reimbursement. They were in Connecticut. So there were many graduate school opportunities there, and they'd pay for it. So I'd work during the day, and then on the nights and on the weekends, I'd do my graduate school work and finished in 6 years, And so, but I wouldn't do it any other way. Just like you, people are like, oh, that must've been brutal. You're like, it was great. I had a great time.
I actually got in trouble.
Exactly right. That's exactly right. Well, what a great story. So tell me a little bit about if you remember the first time that we met.
Yeah, well, it was at Pfizer, right? You were sitting in front of one of those robots and working on the software. I think what it did was just finding the first mass, right? The M+H.
Correct.
And if it didn't find it, it just either redid it or it did a ramp, I think. I can't remember. And then it got the products, and it just did some ramps to get the products. And then that's what you did. You just loaded it up and went on.
It was kind of it was those were in the days of high throughput screening, which I understand some companies still do that. It's not what I see. It's not as popular as it used to be.
No. Nothing came of it.
Nothing came of it. I mean, you would do literally millions and millions of molecules and design them
Mm-hmm.
and not really get any hits.
Yeah.
And so it was a lot of wasted resources. But
It was a good
man,
idea, but
wow, was it fun for analytical chemists? Oh, wow.
yeah.
But anyway, that I remember meeting you, and then so that's wonderful. So
And then after that, it was Salt Lake City Airport, or on a plane going somewhere, or coming back to Salt Lake, right? It's like
that's
every time there was a meeting, where's Shane?
yeah. So Pat at one time was based out of Salt Lake, and I connect through Salt Lake, flying from Lewiston, Idaho, usually. And so we'd often be on the same plane, going to the same conference, sometimes sit in first class together, or we'd
Yep.
sometimes if we had a long layover, we'd actually go outside the airport and eat something.
Yep. Took you to Moochie's one time in Salt
Oh, that
Lake.
was
Yeah,
good. That was
yeah,
really,
yeah.
really
That went on Guy Fieri's show at some point in time too.
Oh, is that right?
Yep,
You know
yep.
what's interesting? I actually, for the first time ever, went to a Guy Fieri show
Uh-huh.
or restaurant in
Right.
Vegas last weekend. Phenomenal. Oh man, and it was so reasonably priced.
Yeah, yeah.
I couldn't believe it, but it was just very unique. So it was
Yep.
really, really good. And that's what I've really loved about science and the industry and bioanalysis and mass spectrometry. It's yeah, I mean, there's been cool tools. You and I could talk about mass spectrometry, we could talk about chromatography, we could talk about all those things, but the relationships that we've made over our
Mhm.
lifetime,
Yeah,
that's the most important part.
yeah,
That's the
yeah.
really, really cool part. So so tell us, you told us a little bit about your own science history. So tell us about owning your own CRO and your LCMS lab. Tell us what that was like. How many years was that? What was the risk that was involved? What was the upside? And get a little bit more detail than you already have.
Well, I think it's very stressful. You know, even with Advion, all of a sudden you realize that you're responsible for the other people. Each
Absolutely.
decision you make, right? And even there, I had Jack Kenyon, or I had partners, right? There was always someone else there, right? Tom Kurz, Jack Kenyon, et
Yeah.
cetera, there. But each decision that you make can mean life or death, right? I mean, it's important stuff. So your decision making changes, and
Yeah.
you have to become much faster at decision making. You
Yeah.
can't dwell on things,
Right.
right? And when you start to see a trend, whether it's your staff or the clients, you need to act on it right away. You
Right.
really cannot take too long. There are definitely things that you think about,
Yeah.
but boy, you got to be really good with decision making. And I
You
think
do.
that's really important, is to be. Someone said, Pat, you jump off the cliff all the time. I said, yeah, I know how I'll land. You know, and you just have to be, what's the worst case? What's the best case?
Absolutely.
What's the one I want? You
Yeah.
know,
Yeah.
then you
Well,
go.
it's interesting you talk about that because you came from Bristol, big company, especially after it merged with Bristol with Squibb, so Bristol Myers Squibb. I came from Pfizer. We both started CROs. I'm still own one, and what was different from me is you would sit in Bristol or you would sit at Pfizer. They would ruminate
Mm-hmm.
on making decisions for months.
Oh, and they'd flip flop, left, right, left,
Right.
right.
Yeah, and that was
Oh, that was yesterday.
yeah, right. That was very frustrating for me because it sounds like you just make a decision.
Yeah.
Now, in a smaller company, what makes you nimble, and that's what's made us successful, is yes, you make a decision, and you got to have that gut feeling. So you got to have that intuition. A lot of people don't have it because they're afraid of risk, and you do. Like, let's just say you have 50 people working for you, then well, you have 50 families to support.
Uh-huh.
A lot of people aren't willing to kind of put that on the line. But here's what I like about a small company and being in charge of it and driving the direction is you can make a decision. If you think it's wrong, make another one.
Mm-hmm. Yeah. Yeah.
Not a, I mean, no decisions are fatal. The
Mm-hmm.
only thing that is fatal is death. And if you
Mm-hmm.
can keep making decisions, they might not always be the best ones, but you learn from your mistakes and you keep making them. And so I'm
Yeah.
totally on board with you. It's
Yeah.
like those decisions are, and it is stressful. It's
Yeah.
stressful to know that your livelihood and everybody else's livelihood is dependent on decisions that you make. And some you can control, some you can't. We couldn't control COVID-19.
Right. Yep.
No,
Yep.
but we can control how we respond to it and what our attitude is.
Yeah.
So Anyway, so tell us more. So you're in the CRO world.
Yeah.
You're probably working. Your kids are sleeping at the lab. They're helping you. All the, I mean, I know I've heard some of your stories. You had to get special security protocols because you'd be coming in this building at 2 a.m. in the
Oh,
morning.
yeah, that was when we were a consultant at DuPont. So yeah, and even Bristol Myers, all the security guards got to know me.
So it's like 2
2
a.m.
a.m. What are you doing here? Oh, don't worry.
Right. It's Pat. It's 2 a.m.
Actually, with St. John's in my graduate school, same thing. I had to let the security guys know who I was, and there were no cell phones, but they asked me to give them a call a couple times. You know, if I was in there at night, just give them a call to let them know I was okay. So
Yeah.
yeah, I forgot about that. I totally forgot about that. Yeah.
Wow.
But
Wow.
yeah, so I think then what happened was we started to grow the New Jersey lab, but the Salt Lake City lab was positioned and ready to explode. And so the CEO, Dennis Lin, asked me to come out and help from a technical standpoint. And so I went out there and ended up moving to Salt Lake City in 2000. So my kids then went to their eighth and ninth schools in 4 years.
But Salt Lake City's a nice place to be.
I liked it. Yeah, yeah. My daughter and grandson are still there. Yeah, Nina and Dylan.
So,
But
so you
so
help in Salt Lake. You
yeah,
basically sell your portion of the CRO, or they buy your interest, whatever it is. And then what happens? And then now you're at PPD. Tell us that story, and then what you do now.
yeah. Well, what happened in between was we got acquired at Tandem. We got acquired by Equity, right? And that allowed us to consolidate. It was a family business. Imagine having 50 shareholders, some of them 3 months old and some of them 80 years old, right? So you
Great.
can't make any decision. Talk about a small company, you're not nimble, right? You need to be nimble.
Right.
So the equity allowed us to consolidate ownership. Then we were sold to LabCorp. I stuck around at LabCorp. I became the VP of business development at LabCorp for the bioanalytical lab, which was interesting, fun. I would never want to do it again, but it was interesting and fun. And then I needed a change. Nothing was exciting anymore. Nothing was interesting, right? And LabCorp actually was quite good to Tandem Labs. So it's surprising, this huge company, right? We went from, you know, 200 people to 20,000 people.
Yeah.
So I started looking for other jobs, and I ended up getting a director of global strategic marketing with Thermo. And what that allowed me to do was, I say, back to school, right? And that was a no lab, right? It was marketing and business development. It was helping new products, right? High resolution.
You know what? I forgot about that stop in your career, but I'm remembering it now.
Yeah, yeah.
I forgot about that, but yeah. So Thermo is they are a vendor that sells mass spectrometers and lots of other,
Everything. Yeah, everything.
including disposables to the scientific
Mhm.
industry, chromatography
And
supplies,
medical. Yeah, they're
and
massive.
medical. Yeah,
I mean, so
clinical.
I went from I think it was 4 billion at the time. LabCorp was about 4 billion. I went to Thermo, which was 16, and it went to over 24 billion by the time I left. You
Wow.
know, so I was there for 5 years. But that gave me, like I said, that allowed me to go back to college, right? I learned more about business, about myself,
Awesome.
about strategy, and
Yeah.
global, right? I was flying all over the world with that company, you
Yeah.
know, 150 to 200 planes every year. You
Wow.
know, one year it was 300,000 miles.
Oh my goodness.
Yeah, yeah.
Wow. Wow. Now, at this point, are your kids out of the house yet when you were with Thermo?
Yeah, it's a funny story. I don't know if they'll ever see this. They'll be embarrassed a little bit. But what happened was they were in their first year of college, I think.
Okay.
Maybe last year of high school, first year of college, and we called them in, said, "Hey, I got to tell you something. I'm going to change jobs." And both of their heads just went down like, "Oh no." And I said, "Don't worry, this time I'm moving." And they go, "Oh, what's for dinner?" Yeah, why?
I'm not surprised.
Yeah,
Oh,
so I got a. It was during the recession, and I was very fortunate. I couldn't have afforded to live in that Bay Area otherwise, but it was during the 2008 recession. It was 2010. Bought a condo in San Jose, and yeah, it was great. I loved it. I loved well, I love the freedom that Thermo gave me, introducing new mass spectrometers and software. All the people I met, just unbelievable. It was great.
That's cool.
But the person that was my manager at LabCorp actually had resigned and went to PPD and became executive vice president of PPD Laboratories. And I think on day one, he had the headhunter, the HR guy, give me a call. And so it took about a year. It took about a year for me to finally leave and go to PPD.
So tell us about PPD, what it does, and what PPD is, what it stands for, all those things, and then what's your role there?
Yeah, so PPD, it's about a little over 30 years old. It started out as several different companies. Then it was part of Pharmaco and some of these other things.
Right,
And
I forgot that.
there's links back. I mean, it's just unbelievable. This would be a whole different discussion. There's always links back in my career to someone that helped create something that I did or joined 10 years later, right? So,
Sure.
I mean, I got my LCMS in Syracuse because of Steve Unger 10 years before me. It was just so many of these connections. Dennis
Absolutely.
Lin, my CEO at Tandem Labs, was a president at one point in time at Pharmaco, right? I mean,
Oh, I didn't know that.
all of these connections, right? All of these strange things, and so a lot of tiny world stories there. But they became pharmaceutical product development, PPD. They were publicly traded. Then probably about 7 or 8 years ago, they were taken private by two private equity groups. And then we just had an IPO in February, so relisted back in the Nasdaq this last February.
Well,
So yeah.
congratulations. I think it's a great story, and it's cool that you're a part of that as well. And so, what's your role there?
Yeah, so I started when I joined. I started the biomarker lab there. So I went from mass spectrometry. So Tandem Lab, small molecule mass spectrometry, right? Then go to Thermo and it's mass spectrometry, large molecule, small molecule, you name it, as long as it's mass spec. Then I go to PPD, and now it's molecular genomics and flow cytometry and ligand binding and immunogenicity, and then mass spec was the last one that I brought in, the last one I started. So I've spent a lot of time there, and what happened was there was a newer, I won't call it new technology because it's not, but there was a new trend starting, right? This is the thing that I recognized way back when. Okay, LCMS is happening. Let me get involved in that. Okay, CROs are happening. Okay, let me go get involved in that. Then I tried to, move history by going to Thermo and help direct things, right? And then went to PPD and started this group. What I learned was the next trend that was really starting to happen was cell and gene therapy. And the new tools for that, or the tools required for that, are flow cytometry, molecular genomics, and cell-based immunogenicity.
You know what? Can you, for those that don't know what gene therapy is, just describe it. Describe what you're talking about. The important types of therapy that can be coming online in the next few. Well,
Oh
it's already
yeah.
here, but it's becoming more popular.
Yeah, we actually, yeah. I've been pretty fortunate with this right from the start. What you typically do is you can take a inactivated virus and modify it genetically so that it can modify the patient's genes so that it can now produce the right protein. So I guess one of the good example might be sickle cell anemia, right? So you're able to change the genes so that now you have a different way. The correct proteins are now expressed where normally they aren't, and you're also able to create what I would call personalized cancer vaccines. So you're able to take out a tumor and then develop a vaccine to your specific cancer, and then send that back to the doctor and make the changes that way. So it's really amazing.
It's
And
incredible. It really, I think it will revolutionize therapeutics for the next 50 years. And
Yeah, and
it's
if you got to study history and everything, right? When you look at biologics, when they first started, I think it was BR96 came through Bristol Myers, and it was a dud, right? But look at where if everyone had said, "Oh, we're just going to stop
right,
biologics," we
right.
wouldn't have the drugs that we have now. And a similar thing happened with gene therapy, right? In the very first one, someone unfortunately died. That was I don't know how long ago, maybe 20 years, probably 1990s.
And
Um,
there were some organizations that said, you know what, we're not getting into that.
yeah,
They were too risk averse, and I totally get it, Pat. And they're like oligonucleotide therapy and so on and so forth. Same type of thing. There's always going to be risk involved, and some of these patients would probably die anyway
right.
because they have cancer or whatever it is, or a very very rare
Rare
disease.
disease. Hmm. Yep.
Yeah. And
Yep.
it's really cool that you're a part of that, that I'm a part of that. And that's what wakes me up in the morning and gets me out of
Hmm.
bed. It really gives us a purpose. Like you probably, I love analytical tools. I love mass spectrometers. I love chromatography. I love the science. And of course I love the business too. I always have since I was young. My grandfather owned dump trucks and construction. He used to talk about business all the time when I was like 8 or 9. I had no clue what he was talking about, but I do now, you know.
Yeah. Yeah.
So, but it's cool to be a part of that cure of just trying to cure disease and trying to help people with whatever their ailments are. And so,
Yeah.
so
Yeah.
that's cool. I agree with the cell therapy and gene therapy that it's really hot right now. And I don't see it going away. Yeah,
Yeah.
there's going to be trials and tribulations,
Hmm.
but I don't see it going away. I
Yeah.
really,
And right now, the rate limiters are the manufacturing, and then some of the technology around the delivery, right? So that's growing, right? All of these things they need to mature, and you need to have another business environment created around it to enable that newer technology to continue going forward, right?
yeah. And
So,
you described it, kind of led me to another question. And I think the good thing about gene therapy, especially as you describe it with, for example, your own cancer, personalized cancer, is it can be very personalized.
mhm,
Highly, I mean, right now, what we do is, and this is just what we have. I'm not saying it's bad or good. It just is what it is. It's like we have prescriptions that work for everybody. It doesn't mean they work great for everybody. It
mhm,
doesn't mean it's optimized dose for everybody. But that's what it is. Here, take 3. I'll give you a very, very crude example. Here's 4 Tylenol. This will help your pain. Well, 4 Tylenol works for some people. It might not work for others.
mhm,
And so, whereas gene therapy can really take that to a next level and be very, very personalized. And so, that's cool that you're in that. You know what's cool about it too is that, and I'm seeing that, and there's some people in my own lab that, again, we came from small molecules. We've talked about that in previous podcasts, but let's just think about a small molecule. It's easier to analyze than a large molecule like a protein. We're just going to leave it at that without any other explanation. So we all came from the small molecule world. So then it's like, well, I guess we have to learn large molecules now.
yeah,
And then people are like, well, now we got to learn about all these other kind of therapies and biology. And yeah, so
biology, yeah,
I
yeah,
mean, it's like
yeah,
what education teaches you, and a mindset of always wanting to learn and really never be outworked. And part of that is learning, is that you can learn anything. You
mhm,
absolutely can learn anything. And that's what
yep,
I'm a lifelong learner. I can tell you are too, Pat. I
yep.
love to learn. I love new information. And you know what? Most people hate to play Jeopardy against me. They keep telling me to go on Jeopardy. I don't know why.
Yep, I love Jeopardy.
Yeah, actually, some are just very useless knowledge, but I like useless knowledge.
Yeah,
So
yeah, definitely.
that kind of leads us into the next one. So obviously, even with gene therapy, you still have a lot of element of analytical tools. You still have to make
Mhm,
measurements. We talked about that last week
yep,
with a SciEx vice president who they're developers of mass specs and mass spectrometers and other tools. And so you have to make a measurement. So that leads me to the next question. What do you think is the future of bioanalysis and LCMS? Because again,
yeah,
I don't think LCMS is going away either.
no,
Liquid chromatography and mass spectrometry. And a lot of people are like, well, now we have these flow cytometry and all these, ELISA assays. They're going to replace mass spectrometry. I don't see it. I
No.
also don't think that mass spectrometry will replace all ELISA assays. I
Yeah.
just don't see that either.
Yeah.
They're complementary.
Yeah.
So tell us about that. What do you see as
Yeah.
the future of bioanalysis
Yeah.
and LCMS and maybe even analytical tools in general?
Yeah. I think you will see the bioanalytical, which migrated from LC to LCMS to then bringing in ELISA and immunogenicity, right? And now I'd say maybe 5 years ago, you started to hear some people talking about flow cytometry. And now because of the cell and gene therapy, a lot of PCR. So the discussions will continue, right? Some of the meetings that we go to and the regulatory discussions, right? They're all expanding. So what started off as a biomarker laboratory for me is now doing a lot of cell and gene therapy support, which I would just, for ease of use now, rather than complicating things, just say it's bioanalytical. It's bioanalytical, right?
Absolutely.
And so
Well, you know,
that's, so I think it'll change that way for sure.
well, you know how it used to be always the small bioanalytical group and the large
Yeah,
bioanalytical group? I think it's just bioanalytical
it's
now.
just bioanalytical. Yeah,
Yeah, because you now have biologists that are running mass spectrometers, chemists that are running mass spectrometers, analytical chemists. I mean, you have different skill sets, which is great.
yeah. And then you've got these surrounding technologies again that I talked about, right? And in order to evolve and progress, you need to have the supporting industries continue to also progress, right? So, sample management, right? Now people are gonna they like over the last 6 months, 4 months, how do you run a clinical trial? Well, maybe the person should stay at home, right? And how can you draw the sample while they're at home? Or maybe they just go to a doctor's office and get the sample drawn. So there's
Stay
that
tuned.
patient centric.
Stay tuned. I'm having Kevin Bateman on my podcast about virtual trials.
Yeah,
And
there you go. So yeah, yeah, perfect. Yeah. And he wore it. He wore it. He had it on his arm.
absolutely, yeah.
Yeah, yeah,
So that's so true. That and that's what a lot of people. We'll get into personalization here in a minute, but so we talk about these analytical tools that we have to use for measurements, and it's just one group now, bioanalytical, right? Not large molecule or small molecule.
mhm,
And I think what is important about that is we think about what a clinical trial is. And a clinical trial is this, and that's what COVID-19 has stopped a lot of these clinical trials because people are not allowed to go into a hospital to go draw their blood because the hospitals are closed because they're trying to deal with COVID-19 or potential COVID-19 patients. So
mhm.
it stopped clinical trials. So what Pat is talking about is how can we move those clinical trials where people are being tested for certain therapeutics to save their lives? How can they stay at home and still be tested? And that's it leads to my next question, but that's where personalized medicine, I think, really, really comes into play and
Yep.
personalizing all of bioanalysis. So that's my leading question. How do you think bioanalysis and chromatography and mass spectrometry or those measurements to develop better therapeutics for people, how will it be personalized in the future?
Yeah, yeah. I think, when you look at let's call it the CDX, right? Companion diagnostics industry, right? It's going to be how can you make these.
Why don't you describe that for the people, what companion diagnostic means?
Yeah, so when you have a let's see what the best way to do it. You want to develop a test that allows in this case it would be a physician to have better confidence or greater confidence that the drug they're going to prescribe will work for that person. The way that they do that is during the drug development, they've done testing of biomarker or biomarkers to show that in this case it works, in this case it doesn't work, or it won't work as well. And it allows them to be more customized in directing which treatment or treatments will work for the patient.
Let's give a really simple example. There's many. Some of them are physical biomarkers, some of them are chemical. Let's think about a chemical biomarker or a companion diagnostic. This is an example, a very simplistic example. Let's just say that somebody is an organization, a company, is trying to develop a drug that lowers your blood glucose. The
Mhm.
simple test is, did
Yeah,
your blood glucose lower? So,
yep.
and you can test that with a blood glucose monitor. That's really, really simple. But
Mhm.
there's that's a biomarker. Glucose is a biomarker in the body. Some other ones are much more complicated, but many pharmaceutical companies, in fact, most, do not develop pharmaceuticals or therapeutics now without a companion diagnostic,
Mhm.
because the issue can be that, and correct me if I'm wrong here, Pat, that sometimes understanding if it works or not can be very subjective.
Yeah.
And you want to try to remove the subjectiveness and make it as much as a scientific yes or no as possible. Would you agree with
And
that?
time function, right? They don't want to have to guess. They don't want to waste time.
Right?
The patient's time, money, effort, right? It's very traumatic in many cases.
It is. I mean, I remember thinking about virtual trials and sampling at home and doing clinical trials at home. I just remember visiting a client, and we were in this near a hospital. And I can still remember waiting in the waiting room like you've done before. You sign your name in, you get a badge, all those things. And I can still remember a taxi cab pulling up and me watching this lady who had her head wrapped up, which I assume she probably had no hair.
Yeah.
She was very frail. She was in a walker, not super elderly. I would say in her 60s, but still had lots of life of potential left, but she could hardly make it into that hospital. And somebody had to come with her, and she had her walker.
Hmm.
I assume that she was coming in to probably take like a blood time sample, like a
Hmm.
48-hour time point, a 72-hour.
Yeah.
That just seems a
Yeah.
little bit unnecessary for me.
Mhm.
So I'm
Yeah.
glad there are going to be other options, and we'll hear about that in a couple of weeks as well
Yep. Yep.
from Dr. Kevin
That's
Bateman.
definitely important. Yeah, that's gonna be a great one. Yeah.
It is. And so I like that. So I like how you're talking about it personalized. What about instrumentation? This fascinates me because I love instruments. How do you think instruments are going to be personalized?
Well, I don't know. It's going to take a long time.
Maybe.
They'll be smaller. It has to. There's always been a push towards smaller mass spectrometers, right? There always has been. How do we get it in a doctor's office? I mean, when I was with Thermo, you heard some amazing stuff using it in the operating room, right? Okay, am I weak?
Crazy. Yeah.
I mean, just the scientists are strange. they are, they're artists, right? When you look at a painting, you're like, how the hell did he think of that, or what made them think of that, right? And it's
Yes.
no different for scientists.
No.
It's no different.
No.
So I think anything is possible. It'll take a lot of time. I think a lot of the migration will come through proteomics, though, right? I think that's where,
Really?
yeah, I think that's
Really
where,
interesting. I don't necessarily think that, but I want to hear your thoughts. I think
yeah,
metabolomics is the way to go. I want to hear your
that's
thoughts.
too hard. That comes after proteomics,
Well,
right?
yeah, it does, but it tells you what happened, because I think it tells you what's expressing your genes. And obviously, this is a great conversation, and I would love to have it, but it tells you how you're expressing them. We can't change our proteome. Well, you kind of can't, but we can change how we express it. But I
Yeah,
want to hear about proteomics, because
yeah.
I think proteomics is way more difficult than metabolomics. So tell me your thoughts.
I
By
think
the way,
it's just a
I had,
puzzle, right? It's the
it
same
is a
same
puzzle.
sequences all the time, right? It's much
I
easier than metabolomics.
Well, you probably know Tim Garrett, right? He was on my show several weeks ago, and of course, he's a metabolomics guy, so you know what he thinks. But I want to hear. I think proteomics is very difficult to understand. I'm not saying it's possible, but
Yeah.
why do you think that's a future?
I think it is because you're really looking at I would say what's the functionality, right? So
Yeah.
so how is this patient responding from a functional level? You know, so I just think it's the next step, right? You couldn't do metabolomics would be meaningless without proteomics and genomics. Proteomics would be useless without genomics. So the
I
order
get.
is kind of genomics, proteomics, metabolomics. That's kind of how I view it.
I understand what you're saying, because without the proteome and the genome, you're dead.
Yeah,
So
yeah,
I totally get that, and
yeah.
so I don't necessarily disagree with you, and it'll be fun to watch. Now here's my view on this, and I'm hoping to have some. I'm in the works of getting somebody on the show that is going to talk about this. But just recently, on a white paper, I think it was in analytical chemistry, Dick Smith's lab at the Pacific Northwest Laboratories, in actually near me in the state of Washington, they actually developed an ion mobility mass spectrometer with the resolution of a normal mass spectrometer or better,
Hmm.
and it has an infinite path length.
Hmm.
And so Now, the cool part about ion mobility spectrometry, does it have issues? Yes, we'll talk about that later. But the cool part about it is, I love mass spectrometers, and I wish we could use them in a doctor's office more easily, or we could
Yeah.
personalize them for home. Here's the problem: you always require a vacuum.
Mm-hmm.
That's the problem. That requires energy, space, lots of money. It's big. Now, the only time they don't require a vacuum is when you use them in space and you vent them outside so that space is the vacuum. And so, but ion mobility mass spectrometry, I worked with Herb Hill's group. He's emeritus status at Washington State University now, but he was big into ion mobility mass spectrometry. I was working with that, 20 years ago. Well, 30 maybe. I don't know. I'm dating myself, but
Uh-huh.
I think that might be the future for analytical tools. But yeah, we'll see. It's really, really cool. So we're getting ready to wind this up, Pat. So I've got to ask this question. I love it. So I want to know about your wrestling career. I'm a wrestling coach. I was a wrestler. My 3 boys wrestle. One of my sons, my oldest one, is a 4-time state champion, 4-time high school state placer, runner up. So all kinds of cool stuff, and all my kids love it. And I think it's a great sport, and it probably put you, and I have lots of friends, of course, who I hang around that are wrestlers. They have the same mindset as you.
Mm-hmm.
Once you've wrestled, everything else is easy. That's what Dan Gable says, and it's true.
Yeah.
It gives you a mindset of you're never going to be outworked. This is nothing. I'm sure you think about your work ethic now, and you work a lot, and you're traveling and all this, and then you think about all those times wrestling, managing your weight, training.
Yeah.
Doesn't even compare.
Yeah.
And not eating.
Not eating, yeah, and then still having to go, yeah.
You'll
I
have to
mean, I used to try it after school. I'd in between work or after work, I'd go run 5 miles every day, every day. Yeah, yeah, yeah.
Tell me about your wrestling career. Like, how old were you when you started? And then we'll get to the Jim Gibbons story.
Yeah, yeah. I wasn't able to start until ninth grade. So first
Really?
year,
Okay.
ninth grade. But then as I was really starting to get, I call it more confident and more aggressive, during practice, my ligaments were torn. So that was the end of my ninth grade season. So
Oh,
that was it. Never got to wrestle a match.
oh no. So then you
Yeah.
came back your sophomore season?
Yep, sophomore, yep. And then I was like 94 pounds and wrestling 98. And then, they didn't really have 91 back then. Towards the end of the year they did. So then I went into the tournaments at the 91. But the first parts of the tournament I went in at 98 and probably weighed about 98. And then 11th grade, I did a lot of more weight training, but I still wrestled 98, but started out maybe 110, 112, and worked my way down. I did really well in my 11th grade. Placed in a couple of tournaments. And, to put it in perspective, just my high school, had over 3,500 kids in it. Just
Oh,
my high school.
goodness.
Yeah. So
Oh,
wrestling on Long Island, Section 8, is hard. Getting out of your county into a state tournament was ridiculously hard.
so just
Right?
making the team was difficult.
Making the team could be hard. Yeah. They're 3 deep, easy. Yeah. Yeah.
Oh my
Yeah.
goodness.
Yeah.
Wow. Do they
And
still
then,
have schools that big in Long Island?
yeah, that school's still there. Yeah. Yeah.
Oh my. Anyway, okay. So
Yeah.
you went to place in a
And
couple
then, yeah,
junior.
then 12th grade, I would say this was not the right thing to do, but I lifted weights like crazy and went up to like 130 pounds,
Wow.
and I still wrestled at 114. Yeah, so that wasn't so smart. And then to get to the end,
You
my
know,
last
you know what, Pat? We didn't know any different back then.
no, no,
We
no,
just didn't.
no.
Our coaches
It's just
didn't.
what we did. Yeah,
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 nutrition was so infantile. I mean, you didn't know about it. So
yeah, yeah.
okay, so you're
So then the last match that I was going to wrestle in high school, they had the Eastern Nationals. I don't know how many people were in it. It was at the Nassau Coliseum or something like that. It was huge, right? I mean, it's just crazy.
right.
And so I'm like, all right, this is huge. And I think there were like 80 something kids in my weight class, and I went up. There was no way I was going to lose weight. So I wrestled up. I think I weighed 114, 115 pounds. I wrestled at 121 or 123. So whatever, I'll just have fun.
Yeah.
And yeah, so I pinned the first few kids, and then went against this other guy. And one of my friends said, "Hey, you're going to go against that kid from Iowa." And I'm like, "Well, who is he?" He goes, "I don't know, but no one's lasted more than 10 seconds with him." So I'm like, "Whatever, okay, that's fine." So I start wrestling with him, and you know, I lasted the longest with him in the tournament. I think it was 27 seconds.
Now tell us who you told me this story before. So tell me who that was.
Yeah, so it ended up being a guy named Jim Gibbons. And Jim Gibbons, just like I did go to Dan Gable wrestling camp in 11th grade too.
Okay.
Jim Gibbons ended up being, an All-American wrestler.
Actually, I thought he was a D1 champ, wasn't he?
Yeah, I think he won Division One. Yeah,
Yeah.
out in Iowa, and then he became the coach and in the Hall of Fame. And if you watch NCAA wrestling now, especially out in Iowa, he'll be the announcer.
He's the
So
Absolutely.
I recognize him. I'm looking at him like, whoa, wait a minute, that's Jim Gibbons. Wow.
Now, so tell us, because I don't know if you want to tell the details, but maybe I thought I heard this from you. How did he pin you? What
Oh
position?
yeah, it was crazy. I don't know. It just shows you how flexible you were when you wrestled, because both of my knees were on the mat, and the last I remember, both my hands were out front, stretching out just so he couldn't turn me. Yet somehow my upper body turned around. So yeah, my shoulders were on the mat, so my knees were also still on the mat. Yeah.
And you didn't hear any snapping sounds from your
No,
ligament.
no, no, no.
Oh my goodness.
Pretty flexible.
So he pinned you while you're on your knees. He was an amazing wrestler. You know what?
Yeah,
That's a great story. I mean,
yeah.
to know that you did that well in a tournament, and you went on and you didn't give up. I mean, you still wrestled at the junior college level, and that's what I tell parents, when they decide if their kid should wrestle or not. Well, he's not going to be any good, and that may or may not be true, but it's going to change your
Yeah.
life and your mindset.
It allowed me to maintain my health and my core. And if someone asked me, go do 25 push-ups right now, yeah, do you want me to put the band on or just 25 straight? You want me to put my feet up?
Absolutely. What's important too, I think, is that, and I'm not saying that, especially back then, that we managed our weight in a healthy way, but we knew no other way. We didn't know any other way. But a lot of parents nowadays, they don't like to put their kids in wrestling because they're afraid of, quote unquote, I don't use this term, but cutting weight. I don't use it.
Mhm.
I call it managing your weight, because you know why? You're going to manage your weight the rest of your life if you want to be
Mhm.
healthy.
Yeah.
And I mean, a wrestler knows what they can and cannot eat to
Yeah.
maintain a healthy lifestyle. And I've taken that into my, adulthood years. And so, Pat, this has been awesome. So I always have my last question. You're probably familiar with it, and you probably have a lot of these stories. So you can tell me just one of your major ones, or you can tell me as many as you want. We have no time limit. Tell us one apparent failure in your life that caused more success for you. Now, and
Yeah.
a failure could be something that you call it's a failure, or somebody else might look in and say, "Oh man, he failed at that." What would that be?
I don't know if how to say it. Is it a failure or not? But I interviewed when I was at Bristol Myers. It was my first job, and you look and you say, what's important to move up in this position in this company? And even though it was probably wrong at the time, and I know it's wrong at the time. I'm looking, young kid, and saying, "Wow, if you want to move up, you got to go work in an office. You got to get out of the lab."
Yeah.
Right?
Right?
So that's what I tried to do. I interviewed for internal sales, to be an internal salesperson, to sell penicillin, right,
Oh,
internally.
interesting.
I tried to get into QA. Wow, I'm so glad I didn't do that. Nothing against QA, but it's not me, right?
Absolutely. It's not you.
Yeah,
Yes.
not me. So I didn't do that, and it was just a matter of a month later that Jack Kenyon came up and did that consulting trip, which
Wow.
led to going to Advion, which that was it. It was the perfect marriage. A startup, mass spectrometry, regulated business, from scratch, getting to do what I had always felt we should do at Bristol Myers, and Jack and Tom are like, well, you know what you're doing. go do it, right?
Yeah.
You know, and bringing on people and setting the culture. Oh, it was fantastic. Yeah,
That's
learned so
awesome.
much, had a great time, met
Yeah.
great people, and so yeah, that was what I would say. It's kind of a failure because I didn't. And in fact, I even know what I said wrong, that I didn't get the job.
Yeah. But there was a reason they didn't offer it to you. I mean, it's something a good friend of mine says is that everything happens for a reason. And you know what the challenging part of that is? In the moment, you think it is the worst thing that happens to you. Like, oh my gosh, you feel rejected, you feel abandoned. And then it's like you look back, it's like, thank goodness.
Yeah,
Yeah.
yeah, yeah,
That's
absolutely.
awesome. That's
So
awesome. Well, Pat, we're winding this up. I'm going to do a little bit of a post introduction here. So again, follow me at drsneedham.com on that's my website. You can follow me on my YouTube channel and find me on Instagram, find me on LinkedIn, find me on Facebook. This podcast will also be recorded, edited and recorded, and it'll be available on YouTube, on Spotify, on SoundCloud, and also on iTunes. And so follow all those platforms there. Thank you very, very much for watching. And Pat, we'll probably see you on an airplane soon
yeah.
or at some conference soon. Thank you for joining me. Thanks for going in the office and giving us a beautiful scene of what
Yeah,
your office looks like in the back. And so thank you very much. And thanks for being a great colleague and a friend.
yeah. Thank you.
All right. Hey, we'll be in touch. Take care.
All right,
Bye.
bye.